So now we're going to continue forward after we've already kind of talked about what evolution is and how these changes happen, but we're going to take it back even further. We're going to get back to the origins of life and the microbes. So in the first video, we'll get into the origins of life, and in the second video for this section, we will get into microbes themselves. So why do we care? There's a lot of questions that people ask, including questions like, how did life begin? It's really one of the largest and most significant questions we can ask ourselves. And with science, we can get a glimpse into that past. So the phylogeny basically is the evolutionary history of organisms. Think of it like a giant family tree with all the organisms that have ever existed, all showing the relationships between them. Using this type of systemic classification, species both extinct and extant, which we'll talk about in just a little bit, we can organize this into one gigantic family tree that goes all the way back. Now, extinct means a species has died out, it's gone. But extant is probably a term you're not as familiar with. You've probably heard the term extinct. Extant, though, means species that still exist. So like we would be an extant species because we as Homo sapiens are still alive. And when we're looking at these phylogenetic trees, we see speciation happening, these speciation events that look like forks in the tree. So basically the things that are connecting the different organisms together. But a lot of the phylogenies we're looking at are often hypotheses, and that means they're subject to revision. So as we learn more or as we find more organisms out there, we revise these phylogenies all the time. Right? This is something that really went through a major change when we started shifting from morphologically based phylogenies to genetics-based phylogenies. The introduction or the massive knowledge we've gotten in genetics has really changed a lot of these phylogenetic trees. But let's look at some of these evolutionary trees and kind of figure out how do we read them. So here we see fish, bird, human, rat, and mouse. And we see they're connected by lines. The further up their connection is, the closer the relative. So the rat and the mouse are very closely related. And wherever they join together, right here, tells us that this was a common ancestor to mice and rats. So both the rat and the mouse have a common ancestor that was the same. If we go back further, we see both the rat and mouse line connects with the human line. And so that's going to be the common ancestor for mice, rats, and humans. And so as we go down, we see that we're adding more organisms into that group that share a common ancestor, but we're also going further back in time. We're getting more and more ancestral. And so when we're looking at these, this is really one of the things, another misconception or statements people will make sometimes against evolution, like saying, well, if we came from monkeys, why are monkeys still around? And looking at these evolutionary trees, I can tell you that we did not come from monkeys, right? Because the monkeys would be in a line like this, separate group. So it's not to say that we came from monkeys, but that us and monkeys have a common ancestor. It's kind of like if you said, well, I didn't come from my sister because my sister's still around. Well, of course you didn't come from your sister. Your sister isn't your mom, but you and your sister share a common ancestor. You and your sister share a mom. And so the common ancestor is like the parent, right? And the organisms that have branched off of that common ancestor are like the siblings. So we can kind of orient these around differently. They don't have to follow this exact format. So in this case, we can spin them around. As long as it remains accurate, it doesn't really matter. Now that being said, we don't see who's more primitive or more advanced. We're just looking at relationships. Who's closely related to who, and that's it. So these phylogenetic trees represent what we call macroevolution. I may have mentioned this in a past video, that evolution in general is just change over time, and that can be on a small scale or a large scale. And the large scale evolutionary changes, which we're talking about making new species, is called macroevolution. When we're looking at just one generation to the next, we're looking at allele frequency changes. We call that microevolution. So think macro on a macro scale, you're on a large scale. Micro, you're on a small scale. So the example we saw for microevolution was the beak size in response to rainfall and how rainfall affected the food source that these birds had. Now, we've been kind of talking about evolution as if it's this very gradual process, but the pace is not always super steady. Sometimes you have rapid diversification due to certain events, and sometimes it can slow down. So it's kind of contracting and constricting back and forth. So there's two ways that are kind of used to describe the tempo of evolution. There is gradual change where we creep along, and so it's very small incremental steps. And then punctuated equilibrium. So instead of going through very small changes, you go through and then you experience a big change. And then you go through pretty stable and then you experience a big change. Now it's important to note that when we're looking at this right here, when we say a big change, we're still talking in the hundreds of thousands of years, if not millions of years. So it's still something that's going to be happening in a relatively slow, quote, fashion, but in terms of the macro scale for evolution, it's fast. There's also something that can happen called adaptive radiation. So we have a small number of species to start with, and they suddenly diversify into a larger number of species. And there's usually three things that tend to trigger this adaptive radiation from happening. Some sort of mass extinction happens because the competition that was originally there suddenly gets eliminated. And so whoever's left rapidly can diversify to fill in the gaps that were lost right beforehand. Colonization events, so if there's a new location and new resources, there's not as much competition, those who colonize the area can rapidly diversify. And then evolutionary innovations. When you get some sort of new innovative feature that increases fitness, that species can rapidly diversify as well. So adaptive radiation, let's look at the colonization event as our example. So let's say we have an island that is completely uninhabited by any major animals. There are plants, there's microbes, but there's no animals. And let's say some birds and some lizards suddenly are able to get to this island. What you'd see is that if you started with like one or two species of birds, those one or two species of birds would suddenly, when I say suddenly, take it with a grain of salt, but would then diversify into many different species, into 5, 10, 20 different species of birds, because there's all these different what we call niches that haven't been filled in. So there's space at the tops of the trees, there's different food sources, maybe there's seeds, there's insects, there's slugs, there's fish, right? Whatever is happens to be there for food, right? Berries. And so each new species of bird can fill in one of those gaps, one of those niches. And the reason that happens is because when you go into a niche that doesn't have any competitors, that means you get to capture all those resources. And so when we go and colonize something like that, we see this adaptive radiation happen because it helps limit competition. You're making fewer competitors that you have to fight against yourself. Now, extinction is when you lose all the individuals of a single species. So you're looking at one species, like let's say we're looking at Homo erectus. It's an ancient human-like species. Homo erectus went extinct, meaning there are no live Homo erectus individuals anymore. They're all gone. And for the most part, these extinctions can happen and it's no issue. We call these background extinctions because there's no particular catastrophic event, just for whatever reason they died out. Maybe they weren't as good at competing, or maybe something happened, some sort of harmful mutation took over. You know, we don't really know necessarily for this particular example. It doesn't really matter. But extinctions in and of itself happen on a regular basis, and they're not necessarily bad. When it becomes a problem is when we experience what we call a mass extinction. A mass extinction is when you have a large number of species on Earth all becoming extinct over a relatively short period of time. And there's been at least 5. Many will say that we are in the middle of our 6th, where more than 90% of animals became extinct. So 9 out of 10 species of animals were gone during each of these. And this is because the environment has changed in a very large way, in a very sudden way. And so basically evolution cannot keep up with that drastic environmental change. And so because that is just bad luck, there wasn't enough time to develop the things needed to survive, right? There was just a really big change that could not have been predicted, could not have been anticipated. And so most species end up dying out. So again, let's go ahead and look at micro versus macro evolution. So microevolution, we're just changing allele frequencies over a few generations. So this is something that usually we're talking about when we're thinking about evolution we can observe in our lifetime. We're looking at microevolution. But the macroevolution is the accumulated effect of microevolution over a very long period of time. Right? In microevolution, we're looking at a few generations. We're looking at months, maybe years, maybe some decades. But in macroevolution, we're looking at thousands, if not millions of years. And with the 2 categories of extinctions, the background extinctions, these just happen. They're at low rates. It's just part of nature. But the mass extinctions, you see where these spikes are. 1, 2, 3, 4, 5. And some would argue that there should be another one right here. With the loss of biodiversity we're experiencing now, this is where a lot of species are gone. So we're looking at percentage of families. That's not even necessarily species, that's families, that's beyond species, that became extinct. We're looking at 1/4 to over half of families becoming extinct. Now, when we're looking at these organisms, we can classify them into one of 3 major groups that we call domains. And the domains are bacteria, archaea, and eukarya. Bacteria and archaea, these guys are prokaryotic, and the eukaryotes are going to be things like animals, plants, and fungi. But no matter which domain you're looking at something that's alive. How do we define what is considered alive, though? Now, we did talk about cell theory early on, and that's part of it, but there's some more conditions we have to get into that are a little bit more specific, a little bit more nuanced. So, in order to be considered alive, you have to meet these following criteria. You have to be organized into 1 or more cells. That's part of our cell theory. In order to be alive, you must use and transform energy. So you have to consume something, so photosynthesis, cellular respiration, things like that. You have to be able to respond to the environment. Now, it doesn't have to be a response the way we think of a response, right? In terms of being able to move or say something. It can be something as simple as opening a flower because the sun is out, and then closing that flower when the sun goes down. All living things have to regulate their conditions and processes, so maintaining what we call homeostasis, or maintaining a relatively constant internal environment. All living things must grow, develop, and reproduce, emphasis on the reproduction part. And then all living things are subject to evolution by natural selection. So basically all living things have an evolutionary history. Now, when we're looking at these conditions, it's not a pick a few out of this category. You have to meet all 6 of those conditions to be considered alive. If even one of those conditions does not apply, it's no longer considered alive. Right? And we'll talk a little bit about viruses a little bit later when we're looking at the microbes and whether or not they're considered alive. Now, one of the big questions people ask is, where did life come from? And while we might not have a specific answer for that question yet, because it is one of those most mystifying and pervasive questions that we've ever asked, we do have some level of answers. One is that we know that the Earth is about 4.5 billion years old, much older than what Darwin's time thought was. The oldest rocks are about 3.8 billion years old, and the oldest bacteria-like fossilized cells are 3.4 billion years old. Yes, we have seen bacteria-like fossilized cells, which is pretty wild. And when we look at the Earth, we see that we have tremendous biodiversity, meaning a diversity of living organisms. There's a wide variety and variability among all the different genes, species, and ecosystems out there. Now, so how did life kind of form, right? And obviously we can't go back in time and view this in action, but we do have a hypothesis about it. So let's go through that hypothesis, which is broken down into 3 phases. Phase 1 is that small molecules had to form containing carbon and hydrogen. So we're seeing the first hydrocarbons. Now, in the early Earth atmosphere, there was no oxygen gas. So you couldn't have anything going through cellular respiration the way we know it today. Instead, we had carbon dioxide, nitrogen, methane, ammonia, hydrogen sulfide, most likely produced because of all the massive volcanic eruptions. So we started to see small organic molecules forming. And there was an experiment done in the early to mid 50s by Miller and Urey showing how phase 1 could have happened. So they set up this experiment, and what they found is just in a few days they discovered that many organic molecules were formed when they tried to recreate how the early atmosphere looked, and they found that they actually ended up with organic molecules including amino acids. Remember, those amino acids are the building blocks for proteins. That's really significant. They discovered 5 of these amino acids at the time, but now that we have more sensitive analysis, we saw that all 20 were actually produced. So there are 20 different amino acids, and in this experiment, if you were to conduct it, you would find all 20 amino acids. So their experiment demonstrated that complex organic molecules could have arisen from the early stages of Earth, right, based on the inorganic compounds that would have been there. So let's look at what that experiment looked like. So Stanley Miller and Harold Urey, they developed this simple 4-step experiment that demonstrated how complex organic molecules could have arisen in Earth's early environment, right? And so they basically have like this atmosphere, an electrical charge, right? This is the model Earth and the atmosphere. They're heating it up. The atmosphere travels through here. There's an electrical charge. We then have condensation of the atmosphere's contents, so we're cooling it. And what they found is as they did this, organic molecules formed, right? And they called this their little primordial sea. And as scientists, we strongly hypothesize this, that there's 3 distinct phases to life coming to be. One was the formation of small organic molecules. Then you saw the formation of self-replicating information-containing molecules. And then lastly, the development of a membrane. Because one of the characteristics of living things is that they have to make copies of themselves. We have to get to that phase 2, where you have to have the formation of self-replicating information-containing molecules. Now, the molecules that contain information are nucleic acids like DNA and RNA. And there's a hypothesis that says RNA probably came first. But why is that? The reason is that RNA can carry information, which we know from looking at transcription and translation, but it also works as an enzyme. It can catalyze reactions for replication. So it has a dual role here. So since it can do both of these jobs, speeding up reactions and holding information, the idea is that RNA came first. Now, is RNA alive? The consensus says no. And the reason being is that it doesn't it's not a cell, right? That was the one of the 6 things that we had to be able to fulfill in order to be considered alive. We also have to be able to respond to environments. We have to maintain internal environments. And those are things that RNA doesn't necessarily do because it's just a single molecule. And then lastly, we get to phase 3, the development of a membrane, enabling us to go through metabolism and creating the first cells. Now, the first cells, quote, the first membrane-bound structures were not true cells. They were what we call protocells, because these phospholipids formed these membranes somewhat spontaneously. The membrane allows for different chemical concentrations on each side, and these membranes can arrange themselves into this bilayer spontaneously, simply because of the hydrophobic, hydrophilic nature of phospholipids. Now, it's not too hard to imagine that a self-replicating RNA molecule somehow found its way trapped inside of a phospholipid bilayer. And that's what we call a microsphere or a protocell. Now, what happens after that? How did we make that jump from protocell to cell? We're not entirely sure, but it's something that's still being worked out. The fact is that we have a lot of information, and while there might still be some gaps here and there, we're filling in those gaps every single day.